This is one of the most commonly used method for purification of solid organic compounds. It is based on difference in solubilities. And next, for adsorbing activated charcoal is used, and if the compound is highly soluble in one solvent and very little soluble in another solvent, then also crystallization is used. And if the impure compound is dissolved in solute in which it is sparingly soluble at room temperature but appreciably soluble at higher temperature, then too this is used. The solution is concentrated to nearly saturated solution, and on cooling the solution pure compounds crystallize out. This is removed by filtration. And next thing we have, repeated crystallization becomes necessary for purification of compounds containing impurities of comparable solubilities. Second is distillation. In distillation we have this is important to separate the volatile liquid from the non-volatile impurities. So it separates the volatile liquid from non-volatile impurities. And secondly, liquid having sufficient difference in their boiling points are also separated by distillation. Example is chloroform and aniline. Example are the examples, and they are separated by distillation, simple distillation. And next thing we have is this diagram is of simple distillation. Vapors of the substances formed are condensed and liquid collected in the conical flask. Next is the fractional distillation. Fractional distillation is used if the difference in boiling point of the two liquids is not much. Okay, simple distillation cannot be used to then simple distillation cannot be used, and fractional distillation is used if difference in the boiling point is not much. Then fractional distillation is used. Simple distillation cannot be used, and vapors of the liquid have with higher boiling point condense before the vapors of the liquid with lower boiling point. Vapors of the liquid with higher boiling point condense before the vapors of the liquid having lower boiling point. And here, to separate the volatile liquid from non-volatile impurities and sufficient difference in boiling point. Next thing, this diagram is of fractional distillation. The vapors of the lower boiling point reaches to the top of the column first, followed by vapors of the higher boiling fraction. Each successive condensation and vaporization unit in a fractionating column is called theoretical plate. Each successive condensation and vaporization unit in the fractionating column is called theoretical plate, and commercially columns of 100 plates are available. Next, an example of this fractional distillation is crude oil in petrol industry. Okay, and simple distillation was aniline. So its example is crude oil in petrol industry. And next is distillation under reduced pressure. This is used to purify the liquids having higher boiling point and the liquids which decompose at or below their boiling point. So this method is used to purify the liquids having very high boiling point and those which decompose at or below their boiling point. Okay, so this is distillation under reduced pressure. Example of distillation under reduced pressure is glycerol from spent lye, and vapor pressure is equal to the external liquid boils at temperature at which vapor pressure is equal to the external pressure. Glycerol can be separated from the spent lye in the soap industry by using this technique, glycerol from spent lye. And this is given simple packed column, bubble plate column, and glass beads are given. Next thing we have is distillation under reduced pressure. A liquid boils at a temperature below its vapor by reducing the pressure. Okay, so next is steam distillation. So the steam distillation example is aniline water mixture. Aniline water mixture is a steam distillation, and in this technique is applied to the substances which are steam volatile and immiscible with water. Steam volatile and immiscible with water. and P is equal to P1 plus P2, this thing. And next we have differential extraction. This differential extraction, when organic compound is present in an aqueous medium, it is separated by shaking with the organic solvent in which it is more soluble than in water. When organic compound is present in aqueous medium, it is separated by shaking it with organic solvent which is more soluble than in water. If the organic compound is less soluble in organic solvent, a very large quantity of the solvent would be required to extract even a small quantity of compound. This is called continuous extraction. And next we have chromatography. A chromatography is important technique exclusively used to separate the mixtures into their components, to purify the compounds and also to test the purity of the compounds. And chroma means color, and in this method stationary phase may be liquid or solid, and the compound of the mixture gets gradually separated from one another. The moving phase is called mobile phase. And based on principles involved in chromatography, there are two types: adsorption chromatography and partition chromatography. This diagram is of differential extraction based on the difference in solubilities. And this column chromatography and thin line chromatography are types of adsorption chromatography, and paper chromatography is type of partition chromatography. Adsorption chromatography, it has two types: column and thin line. Adsorption chromatography uses silica gel and alumina, and it different compounds. It is based on the fact that different compounds are adsorbed or on an adsorbent to different degrees. And next is column chromatography. We will study. In column chromatography, separation of mixture over column of adsorbents or stationary phases packed in a glass tube. And in thin line chromatography, we have thin line chromatography TLC, uh, which involves separation of mixture over a thin layer of adsorbent coated on a glass plate. Thickness is about 0.2 millimeter, and adsorbent is silica gel or alumina is spread over the glass plate. This glass plate is called thin layer chromatography plate or chroma plate. And next we have is this RF retardation factor. Retardation factor is the distance moved by substance from baseline upon distance moved by solvent from baseline. Distance moved by substance from baseline upon distance moved by solvent from baseline. Substance upon solvent. Substance upon solvent, x upon y. And this diagram is also given: that jar, baseline, adsorbent coated on the glass plate, simple dot. This was thin line chromatogram. Next, yes, depending upon the degree of adsorption and separation takes place. Adsorption and separation take place. Yes, the relative adsorption of each component of a mixture is expressed in terms of retardation factor RF. The relative adsorption of each component of the mixture is expressed in terms of retardation factor RF. Sometimes an appropriate reagent may also be sprayed on the plate. For example, amino acids may be detected by spraying the plate with ninhydrin solution. And the spots of the colorless compounds are invisible to eyes, but fluorescence in UV rays can be detected by putting the plate under the UV light. And another detection technique is to place the plate in a covered jar containing few crystals of iodine, and it will show up the brown spots. Okay, in next video we will look at the partition chromatography. Thanks for watching.